All right, recording in progress. Um, Good evening, everyone. It is my pleasure to introduce tonight's speaker, Dr. Sharon Eversman. Sharon Eversman is a retired biology and botany professor from the Ecology Department at Montana State University. Her research area specializes in lichens. Her research area specializing in lichens led to an interest in communication between organisms, especially in the plant world. She is a longtime member of the Montana Native Plant Society Valley of the Flowers chapter, and she is currently working on long-term project to control spotted knapweed on hillsides in Bozeman and returning native plants to the sites. Thank you so much, Sharon, for joining us this evening.
Well, hi, everybody. I can't see you, but I'm going to assume you're there. So here's where I'm coming from this, with this. I have in my world now people who are not scientifically oriented at all, and they did read the book on the German, the Secret Life of Plants, and then came out searching for the mother tree, and they said, wow, is that right? Trees talk to each other? And I said, well, not exactly. So let's talk about this entire field of how plants communicate. So some of this is really obvious. So we know that trees, plants communicate chemically. So for example, let's talk about scent and color with pollinators and with seed dispersal agents. So in order to do that, they have to smell good, taste good, look good. So that's one kind of communication. At the same time, they usually have to repel herbivores and pathogens. So part of the plant is attracting people, other organisms, and also repelling things. So more communication. So now there's communication with each other, with predators, and with symbionts. So I know that you're familiar with the one bad apple spoils the group because of ethylene, which is an aerial hormone. Okay, it's communicating. In the real world, they're trying to synchronize all the ripening of the fruit. So all the seed dispersal agents come at the same time, disperse the seeds, and get on with the next generation. So generally, communication. So here's what happens in the lab, and this is an old test on how chemicals travel between plants. So you've got plant one. These are not connected underground. You've got plant one that is chewed on, or a fungus or something, and that plant produces more jasmonic acid, which is a messenger molecule, and phytohormones. Yes, plants have hormones. They move through the plant, they turn on genes, then the plant number one produces defense compounds. Then those defense compounds travel through the air, and they make defense compounds in the second plant, which deter insect predators. Okay, so one plant can signal another one and say, hey, I'm getting chewed on. You produce some defense compounds and you'll be safer. Okay, then this is a sidebar, and it's newer and it's kind of cool. There's a field now called phytoacoustics. And with more sensitive sensing equipment, they have found that the insects, for example, chewing on a plant make sounds. So think eating a salad. And that sound is detected by plant number two, and plant number two makes defense compounds. Cool. So phytoacoustics, a relatively new field. So another sidebar here. So a couple of little journals I've been reading have talked about the sounds that plants make when they're stressed. So let's talk about the transpiration stream. And we know that when light occurs in the morning, the stomates open, evaporation occurs, water evaporates, and CO2 goes in. And then in order to keep the water flow going, there's this consistent stream from the soil through the roots, up the stem, into the leaves. Okay, that's the transpiration stream. And when there's enough water in the soil, that's absolutely ideal. Okay, drought. not enough water in the soil. What happens? You still get the evaporation, but there's not enough water to fill up all the xylem cells that are going up and down. So what happens is that that stream of water breaks, and there's actually an audible sound when you have the hydrogen bonds between the water molecules breaking, and then the hydrogen bonds between the cell walls in xylem, tracheids, and vessels, that breaks, and there's an audible sound. And that is an audible click that evidently can be heard by insect pests. Is that possible? So I'm going to tell you my experience with some evaporation. I was sitting on a big flat rock in Canyonlands one spring. It had rained overnight, and I was sitting on a big flat rock. Sun was coming out, and there were little depressions in the rock, and I kept hearing that. No insects, no bugs, no nothing around. I think I was hearing water evaporate. So until there's a better explanation, that's what I'm going with on why stress plants can attract pests audibly. Well, back to the stuff that's proven. So acacia trees in Africa are known for being eaten by giraffes. So when they're nibbled on by the giraffes, they produce tannins. So tannins are something actually that's in our pine trees all the time. And what tannins do is interfere with digestion of proteins. So animals can actually die of starvation with a stomach full because they can't digest the proteins. So what these acacias do is put out ethylene. The ethylene can travel at least 50 yards. And then other acacia trees can produce defense compounds that are stimulated by the ethylene going through the air. So what the giraffes do, they go upwind. But anyway, this is well documented in Africa. So we've got communication through the air. So now let's talk about underground. And this is where things get pretty interesting. So underground studies in the labs, you've got these plants on the right, the plant number 1, attacked by caterpillars or aphids or fungus, or you can clip it with a scissors, whatever. And they produce organic compounds that travel through the air, signal plant number 2, and plant number 2 has these pathways that then lead to defense compounds. And in addition, now we've got plants that are attached underground by CMN, the common mycorrhizal network. So we have now roots able to communicate. So they can go via the mycorrhizae, the fungi. So they know that there's signaling through the mycorrhizae. There's chemical transport, liquid film, electrical action potentials, all kinds of explanations for how things communicate underground. So this is what we see: trees, bushes. This is what we don't see. So underground, there's this tremendous fungus mycelium that is there, that is invisible unless we start digging. So little fungus biology here. We know that fungi need to get their nutrition from other organisms. They're like animals. They need sugar compounds from something else. So vocabulary, the mushroom caps, that's a reproductive stage that produces lots and lots of spores that are everywhere. And then individual threads that we have here are called hyphae. One is a hypha, many are hyphae, and then all the fungal hyphae together make a mycelium. So what do fungi do? Well, some decompose, like dead things in the soil, dead logs, like oyster mushrooms, molds, like in the refrigerator, mildews on wet towels. Okay. Some are parasites. They feed on living things, make diseases on plants and animals. We have a whole plant pathology department that specializes on fungal diseases. Some hook up with algae, and that makes lichens. And that's how I got into this whole thing in the first place. And here's tonight's major subject, mycorrhizal. The fungi hook up with green plants. So some of this is completely review, but let's just review it anyway. This is a mutualistic symbiosis between a plant and fungi. So what do the fungi get? They get sugars from the tree, from the photosynthesis. So we have sugars that are going down up here from the leaves through the phloem down into the roots and then into the fungus. So the fungi, I mean, I'm not 100% sure of this, but in lichens anyway, what the fungi do is change those sugar compounds from the algae, from the green plant, into compounds that cannot go directly back into where they got them. So once those sugars get into the fungus, they're there, and then the fungus uses them. And then these mycorrhizal attachments that we're talking about are only present in the youngest, smallest roots. You have to dig down and find the youngest, littlest roots to find these combinations. What do the plants get? They get a whole lot. Roots are okay. Roots are pretty good at getting nutrients and water from the soil, but fungal hyphae are smaller, and they can get into little spaces between soil particles. They can make more direct contact with the soil particles, and they can get more water and they can get more minerals. So they're more efficient, basically, than plant roots. So not only do they help with nutrition and water, here's some other things. They can also protect plants against soil pollutants, and this is a specialization kind of deal. They can protect against heavy metals in the soil. Kathy Cripps worked with this over at Anaconda. They can help with root parasites. So instead of parasites, you've got these symbiotic relationships. They can help protect against soil grazers, like little nematodes and other critters in the soil. And this is important. They can get more water than plain roots can get. So basically, the fungi can be very specialized, or they can be generalists. So in nature, about 80% of all plants are mycorrhizal. Basically, if you see a perennial, it's mycorrhizal. So what plants are not mycorrhizal? Generally the annuals, annual grasses, annual forbs, garden plants. So why not? Why are garden plants and some of these annuals not mycorrhizal? Well, two reasons. In gardens, you frequently fertilize. You can fertilize anything, and if you fertilize it, it says, heck, why should I spend my sugars on fungi? I'll just get my whatever I need from the fertilizer. Number two, it takes time to establish these mycorrhizal relationships. So Kathy Cripps has worked with a mushroom and whitebark pine in the lab, and she says it's between 1 and 3 months to establish a good mycorrhizal connection between the perennial plant and the fungus. So let's look at this kind of complicated plant community, and we'll come back to this. We've got trees here that are connected with the mushrooms right here. We have trees connected with trees. We also, they're discovering things like pine drops, Coral oryza. They used to think they were parasitic on tree roots. Now they think, no, they're hooked into the mycorrhizal relationship. So they get their fungi, they get their sugars from the tree, but also through the mycorrhizae, not directly from plant roots. And then we have another couple of systems here. We have this forb and grass connection here that is different from these over here. Now we're also discovering you can connect conifer trees with deciduous trees with, as I said, maybe with these parasitic plants. So we have these two distinct systems. Definitely the trees and mushrooms are one system. The forbs and the grasses are another one. But of course, this is biology. So guess what? It gets more complicated. So later. So let's talk about the two different kinds of mycorrhizal relationships. So if we think about cedars and junipers, they're different from pines and firs and spruces. And then maple, ash, dogwood, sycamore, other deciduous trees have one kind of mycorrhizal attachment. Grasses, ferns and fernalize, they're also discovering some liverworts and mosses, maybe have these kinds of mycorrhizal relationships. And then when we see wild plants, when we see wildflowers, they usually are mycorrhizal. So most forbs, and forb is a herbaceous plant that's not a grass. So what happens with this kind of relationship is fungi actually grow into the root cells. So these are microscopic views of, this is a scanning microscope view, other microscope views that show the fungi inside root cells. This diagram is important because it shows something, that when you've got this good relationship between the plant and the fungus does not break the cell membrane. If you break a cell membrane, basically the cell is dead. So what happens is they invaginate the cell membrane, and you can imagine what the surface area is here, which makes for really good transport between the fungus and the plant, and the fungus and the plant and the fungus. Important point. So recognition, this whole field of cell-to-cell recognition is a huge field that I wish I knew more about. But evidently the fungi recognize the roots to attach to because of a secretion of a plant hormone called trigolactone. So what the plant hormone does is say, hey, I'm here, but it also stimulates the branching. So this branching increases the surface area for the nutrient transport a whole lot. And then, of course, the genes of each symbiont are turning on to say friend, not foe. So we have evidently with these arbuscular means tree-like, so branched, about 130 species, they say, of these endomycorrhizal species. And if you go way back to the colonization of land, you know, in the Precambrian, they probably helped plants live on land. So those are the endomycorrhizae, the fungi inside the root cells. The other kind are the ectomycorrhizae, EM. So these are all of our conifer trees, plus aspens, birch, oaks, willows. We'll come back to that. And then the fungi do not penetrate the root cells. So this is a cross section of a tiny little root. This is the conducting tissue right here. So water comes in through here and goes through these cells and goes up the xylem here. And then sugars come down the phloem right here. And then there's this transport of water and nutrients. And then the mycorrhizal fungi are surrounding the cells. They're not penetrating, they're surrounding the cells. And then they form this network around the itty bitty roots, and they look like this. So if you're familiar with a grass root, you know it's really fibrous. It's really tiny parts. Here's the clubby part of a mycorrhizal root in a like a pine tree. So look at 6,000 species of ectomycorrhizal fungi. And this gets very complicated. So more than one fungus can colonize one tree. Many fungi can colonize one tree. It's complicated to say the least. Then something else that I just have been looking at, some trees have both types. So we have maple and alder, ash, cottonwood, willow, oak, tilia, and even some pine that can have both types. However, they tend to specialize in one type over the other. So you can count on a pine tree having this kind, but maybe sometime during the year, it might have some endomycorrhizae. I mean, that complicates things a whole lot. So biology, no absolutes. So there also, if you think about two other kinds, one kind of mycorrhizae is combination of ecto-endo mycorrhizae in the Ericaceae in like blueberries, huckleberries, whortleberries, and then orchids also have specialized mycorrhizae. So let's talk about a really classic picture here of the network of a little pine seedling right here, and look at everything that's underground from this. So we've got the roots here, we have the mycelium here, we have connections every place. So here's what's going on underground. Lots of fungus mycelium, and this evidently is visibly different in different mycorrhizal systems. So an expert like Kathy knows the difference on these. So here's some literature on this, and the literature is overwhelming. So what plant chemicals could be involved in establishing this association? Well, terpenes, think the smell of a pine forest. Flavonoids, that's a kind of thing that attracts rhizobium to legume roots, for example. Plant hormones, depends on the plant and the fungus species. So the plant exudates from the roots first attract the right fungus. So both the fungus and the plant release signal compounds. Compounds stimulate genetic changes in both the plant and the fungus, and that would be individual. And then what they're also discovering with roots, root-root combinations, is cell walls can change structure and composition. We usually think of cell walls as being pretty inert, and this doesn't seem to be the case. So if we look at a cell membrane, you know, you've got the lipid bilayer here, you've got all kinds of proteins and all that. Typically, from what I first learned, is it's proteins and glycoproteins, polysaccharides that are involved. Polysaccharides and proteins are primarily involved in recognition molecules. So, you know, these recognition molecules recognize compatible fungi species, and recognition molecules in the fungi recognize compatible plant species. And what you might also think at the same time, some other recognition molecules are recognizing enemies. They're recognizing pathogens. So it gets really interesting underground there. And then when we think about fungus tree associations, some fungi really are specialists. They're only with one kind of tree. On the other hand, some fungi are generalists. They can associate with many tree species. And vice versa. Some trees are maybe only one kind of fungus, and maybe some tree species say, you know, any fungus is good for me. So on a practical level, Kathy Cripps has a book out, has had a book out, if you're interested in mushroom hunting. And where do you go for certain kinds of mushrooms? So, for example, lodgepole pine and chanterelles, swillis and whitebark pine. So this book is really good if you're into mushroom hunting because of mycorrhizal associations. So that brings us to this cutesy little thing called the Wood Wide Web, not the World Wide Web, the Wood Wide Web. And this was a term that was actually coined by, I think, the a journal in England when a paper was submitted on how the plant species interact with fungi. So the Wood Wide Web takes into consideration all these communication patterns that are going on in a forest. So we know that there's aerial communications, and it seems to me years ago I saw a paper on how willow talks to other willow, but I haven't been able to find it since. So we know that there's some aerial communication, but the biggest deal is in the soil. So we have compounds that are moving between and among the plants and the fungi. So what's moving? Water, for sure. Inorganic compounds, especially nitrogen, phosphorus, and potassium. Carbon compounds, sugar. Electrical signals, defense compounds can move between all of these members right here. So some images to not have. If you go to this kind of thing on Google, you'll find pictures like this. No, no. So how do you trace the flow of these things from one plant to another? They usually use radioactive isotopes. So carbon-14, carbon-13, radioactive nitrogen, radioactive phosphorus, they put it in one tree, usually in the leaves, in the case of carbon, because CO2 goes into sugars, and then nitrogen and phosphorus can be given through the roots. And then they trace with a Geiger counter from one organism to another. Okay, from one organism to another, depends on where you put the compounds in. So you can use Geiger counters in the field, and Suzanne Simard did that in British Columbia and in Oregon. And then, of course, the best way to do it is the expensive way, and you gather the tissues, plant, root, whatever it is, the leaf, and give it to the lab for lab analysis. So one of the more famous books that came out a 3 years ago was Searching for the Mother Tree. And Suzanne Simard first was in Oregon, at Oregon State, and then in British Columbia. And she was studying the forests primarily in British Columbia. So this is a long book, and there's a lot of biographical information, but she tells about the tests that she did in these forests to try to convince foresters that you shouldn't clear cut, number one, and you shouldn't get rid of everything, number two. So she was in an area where she's starting to feel guilty about clear cutting everything, and started looking at some relationships. And these are just a couple of her many things that she looked at. She looked at alder and their fungi and ponderosa pine. So this combination, and also birch and fungi and Douglas fir. So what she found was this: in both cases, movement of water and minerals traveled both ways. So think about the season of the year and what we have when everything flows. In biology, usually it goes from a higher concentration to a lower concentration. So a higher concentration is called the source, lower concentration is called the sink. So if you think about Douglas fir compared to a birch in the fall and the spring, Douglas fir has more leaf area than birches do in the fall and the spring. So there's more water and mineral movement from the source to the sink, from higher concentration in the conifer to the lower concentration in the birch. In the summertime, there's more leaf area in the birch, so you have more movement of the sugars from the birch to the Douglas fir. So again, this movement depends on the time of year. What she found, and this is just summarizing she found even more than this: the conifers grew better and healthier for more years, and she did this for 3 or 5 years, when connected with the alder or the birch. So alder has nitrogen fixing little things called frankia that fix nitrogen, which means alder has nitrogen to spare, so it can transfer nitrogen to the conifer. And then, because maybe the conifers were healthier, there was less predation by rabbits on the seedlings, on the small trees. And armillaria is a really nasty kind of root parasite on conifers. So when these conifers were, both the ponderosa pine and Douglas fir were connected with the birch or alder, they had less infection with the armillaria. More nitrogen, they were healthier, they were happier. And then another thing to really take into consideration, I would say maybe in our forests and as climate change happens, mycorrhizal connections are really, really critical in drier habitats. You know, those little fungi can get more water than just roots. So water availability is much better with mycorrhizae than with roots alone.
Sharon, we have a question in the chat.
Okay.
Um, Beth is asking, are soil amendments or fertilizers with mycorrhizal effective, or is it just a marketing scheme?
You know, we played with this a little bit on our project, and what they don't know. Basically, the answer is they don't know. Now, soil amendments, I think, are a little bit different than mycorrhizae, but if you add mycorrhizae, what they say is you don't know if they're competing with mycorrhizae that are maybe already present. If you have soil that have no mycorrhizae at all, which is probably possible, that's what I was assuming on our south-facing hill, adding can't hurt, but they're not sure how much it helps. So the answer, it depends exactly on what you're working with. That's the reading I've done related to the project that we've had on our hill.
Great, thank you for answering that.
Yeah, I hate to give it depends answers, but it depends. So worldwide, this mycorrhizal study thing is not limited to North America. So for example, in Israel, they had in the lab, they took soil from the field and they showed transfer between pine and oak. In Europe, I think it was in Finland, they put C13 into old spruce, and this 5-year study in the field found the C13 in the small roots of beech, pine, and larch. Okay, interspecific things here. In Canada, they defoliated some Douglas fir, and that fir increased their own defense enzymes, and then those defense compounds were transferred into a ponderosa pine. What they said was, you know, it could also be electrical signals. They weren't sure if it was a compounds directly or the defense compounds or the electrical. So these are just the trees. And then, of course, there are lots and lots of studies with arbuscular mycorrhizae in the field of grasses and wildflowers. So again, this similar stress reactions seem to be transferred between stressed and non-stressed plants. Non-stressed plants increase their defense compounds. And that, again, that's with the endomycorrhizae. So criticisms, of course, if you write a paper, you would expect to have criticisms. So in most studies, things were attributed to the mycorrhizae. However, the critics would say, but what about movement of minerals through directly between the roots? What about roots touching roots, all that? Is it possible, especially in field studies? So in labs, you can really control things. Field studies, you can't control things as tightly. Now, here's a study that was done in Alberta that illustrates the complexity of all of this. This is a 30 by 30 meter plot of land. It's a Douglas fir forest, and these are all the little Douglas firs right here. So they did the DNA of all the individual Douglas firs. Then they collected wherever they could find little rhizopogons, and they did the DNA of rhizopogon. So this is the North American cousin of the truffle that is so yummy from Europe. I don't know if these are edible. I don't think so. Anyway, all these little black dots indicate where they did the DNA studies of rhizopogon. And then they made this network of things, of connections between these trees. As you can see, it's very complex. So you can see that not every tree is connected to every other tree with the rhizopogon. We don't know what else is in there. They didn't study that. The big deal that they came from that was the biggest trees had the most connections. Little trees maybe had one. This little guy might have had a connection with a different kind of fungus, but not rhizopogon. So we don't know. This was only rhizopogon. Anyway, it gives some idea of the complexity of what they're trying to study in the field between the trees, and this is just 2 species of rhizopogon. So again, the criticism of this, there was no evidence that this is a complete line of connection. They can assume it was by the DNA studies, but we could have the critters chewing on this, and maybe it wasn't a direct connection after all. So my main takeaway from this is studying in the field is difficult. Now, Susan Simard came up with the idea of a mother tree concept. You know, we've just seen this connection with the largest trees having the most connections. So if you've got a forest or even part of a forest with the largest trees, those oldest, largest trees would have the largest number of mycorrhizal connections. And then there was some evidence that there was preference in moving nutrients and water transfer from a mother tree to seeds that were genetically more similar, called kin. So this is a principle actually in biology called kin selection. You tend to take better care of individuals that you're genetically related to. You tend to take better care of your kids and your cousins than you do the strangers down the street. So in this case, there was more nutrient and water transfer to kin than from not kin. So again, that of course was criticized, but here's a couple of points made. Mycorrhizae do not grow to a tree. The tree has got some mycorrhizal connections, and if these little guys tend to be in the same network of the mother tree, it still is going to be, this kin will be preferentially treated to not kin. So this was not exactly repeatable so far. However, what they've discovered that in lab studies, roots can recognize kin and not kin. So again, maybe it's partly roots and partly fungi that account for this mother tree preferential treatment of kin. Implications for forestry. And of course, you know, you're dealing with lab studies and you have to say, well, it worked here, maybe not there. Anyway, almost intuitive kinds of feelings about this say this, that if you're cutting, the difference between a clear cut and maybe not a clear cut is that you're leaving some diversity here, that maybe you're leaving some nitrogen fixers and you're leaving mycorrhizal connections, which will give protections from pathogens and herbivores. That's the biotic advantage of that. So if you're removing all of this, you're removing any kind of nitrogen fixer, including even, well, we won't go with the legumes at this point. We'll just talk about something like alder. But mycorrhizae cannot exactly live in this soil indefinitely. The fungi spores will disperse or become non-viable or whatever. It's going to be really much harder to reestablish any kind of connection here. If you've got plots of stuff left like this, then, you can have seedlings come out from here and the mycorrhizae grow with the seedlings, and you're better off for revegetation to have something like this for reforestation. So, when I think about this, I think about looking southeast of Bozeman. There's the old T that was a ski area a long time ago. It was the Bear Canyon ski area. I've been watching that for almost 60 years, and it has changed very little. That you see a few more trees each year, and it's growing in from the sides. So the T is gradually getting smaller, and a few isolated little trees here and there. But, maybe if they left some trees there, maybe it would revegetate better than it has. So the idea here is you leave large old trees, partly because if they're big and old and happy, they're going to be best adapted for that habitat. So any offspring they might have might be genetically adapted for that particular habitat. So the difference between this and this is huge biologically, to say nothing about erosion protection and spring melt. So, where you've got shade, the melt is much slower. So something like this is going to have much more advantageous spring melt than something like this. So biological, biotic, abiotic. So actually, you're all aware of the problems with whitebark pine all over the Northwest. So Kathy has worked a lot with a particular mushroom called swillis and whitebark pine. So in the greenhouse, she has inoculated the plant seedlings, the tree seedlings, with this swillis mycorrhizal fungi, and they're replanting. That's probably the best approach. So I've seen these pictures of kids with backpacks with little spruce seedlings or pine seedlings or whatever. They dig a little hole, they stick a pine seedling in, say, hey, good luck. If they're not inoculated, the chance that they're going to survive is really slim. Eastern Montana, thinking back, I spent some time in coal strip around the coal mining there. And what they did, the state mandated that they reforest with ponderosa pine. So there was a greenhouse, I think in Corvallis, Montana, where they were inoculating ponderosa pine seedlings with mycorrhizae. And the pine seedlings were then planted already with their mycorrhizae. I'm assuming that worked better. I haven't been back to find out. So some conclusions on all of this, on communication. Plants cannot get along without ectomycorrhizae and endomycorrhizae. You have to have them. When they talk about communication, it's not going, hey, how are you? It usually means a transfer of resources, whether it's water, carbon, as in sugar compounds, other nutrients, nitrogen, potassium, phosphorus, distress signals, and they move from source to sink. I don't see active transport here. I see things moving from a higher concentration to a lower concentration, and it's usually underground. And then the resources that increase, that are better, are used for increased growth and survival. Keeping in mind, maybe there's also transfer between trees because the roots are close together. And maybe the soil is kind of good, that it's moist enough that things can move through the soil, through the water. We don't exactly know what bacterial communities do. There's evidence that there are electrical signals between roots, between trees. So maybe it's not always just mycorrhizal connections, but I'm not sure I want to minimize mycorrhizal connections. And then usually you've got trees close together. So these trees over here might have some really good connections. We don't know about this guy over here. Maybe he has something else over here that connected to, but not necessarily here. So we've got these connections between trees that are close together, but maybe not through the whole forest stand. And then something always to remember, lab studies can frequently demonstrate things where you can control everything and you can see things and measure things that may or may not be observable in the field. So you can control the species strains of what you're working with, the light, the temperature, the moisture, the soil. Field observations are very difficult, to say the least. You got bears, you got rain, you got snow, you've got all kinds of other problems that you can't deal with. So anyway, my usual leaving off plant idea with biology is you never say prove something. You say it's generally true or usually true, or in these conditions, something is true. Never or always, not in biology. So some references. If you want basic biology, botany books. Montana Native Plant Society has some preserved YouTube pages. Kathy Cripps has a great presentation on mycorrhizae. Diana Six did one on whitebark pine. Mark Kreider did one on aspens. So here are these two kind of popular books. Hidden Life of Trees was written in Germany, and primarily, as I recall, it was deciduous trees. And it bothered me a lot because I thought it got a little bit anthropomorphic, and trees are not people. Finding the Mother Tree, interesting. Here's the mushroom book. So if you're a mushroom hunter, you know where to go to look for things. Braiding Sweetgrass is classic when you start thinking about interactions and what marvelous things trees and plants are. I understand she has a new book out called Serviceberry. And I don't know if it's our Serviceberry or an East Coast Serviceberry. I don't have the book yet. This was kind of a cool book called The Arbornaut, where this young woman decided she wanted to see what lived at the top of tall trees. So they built platforms and all kinds of hoists to lift people to the tops of tall trees to find out what's up there. Actually, there was a study in Oregon. I think Bruce McCune did it with the tall trees to find out what lichens lived at the very tops of trees. So the very tops of trees and tall forests are different. And then this is just, preaching to the choir here. The idea is get kids outside. Let them know what's there, what's outside. So basically, that's my spiel. So little end points here. So questions, comments? Okay, I see some. Let's see if I can check the chat.
Thanks so much, Sharon. Oh, that's great information, Carolyn, that Robin Kimmerer will be speaking at the Kootenai College on Friday. Wow.
Okay, no questions?
Sharon, I'm curious. I know that you said that it was your research in lichen that led you to getting more involved in mycorrhizae. How how did you first how did your curiosity first spark about lichen, and do you still go down that road?
Not as much as I used to, because to identify lichens, you need microscopes and chemicals. And I kind of don't have access to those like I did. But in botany classes, you learn about lichens and mosses. And I was Mrs. Nature at my daughter's campfire camp one year, and there's wonderful boulders up Highlight Canyon, and they had lichens and mosses. And I started thinking, I bet those things have names. So I started collecting them, and discovering it was really hard to get to the names, but I got some help from people in Colorado and Wisconsin, and then used what keys were available and just started getting interested because they were kind of cool, on the trees, on the rocks, in the soil. But that led me to thinking about interactions between species. So, lichens are algae and fungi. Mycorrhizae are plants and fungi. There's got to be some similarities. So that led me to all kinds of other kinds of interactions too, pollinators and seed dispersal agents, nitrogen fixation, all the connections in animals. You can talk about the microbiota in the gut. I mean, the species interactions are just an incredible thing to think about.
Does anyone else have any questions to enter into the chat? You can also raise your hand or just unmute.
Hi, Sharon. This is Robin. I just want to say that I agree with you. I really loved Simard's book. It was wonderful. But even in her book, or maybe it was in one of her presentations online in a YouTube video, she says that she uses anthropomorphic metaphors on purpose to try
Right.
and get people to pay attention to the importance and the preciousness of our forests and that sort
Right.
of thing. But I think I work with a lot of herbalists and naturopaths, and we want to believe so badly, that the plants are talking to each other and talking to us. And they don't want to hear that, no, there's no ears, there's no nervous system there, and I think that, yeah, I gave a presentation one time on kinship in plants, gosh, years ago. And it's fascinating, the information I found. And yes, a lot of it is the mycorrhiza. But yeah, there's really, there's compounds that are going between plants that induce resistance, that send VOCs, and that sort of thing. And there is a certain kin discrimination. Here, I'll put this, let's see. I think this is the name of the paper in evolutionary biology. I'll put it in the chat. Or no, wait, that's not it. I'm sorry. Let's see, let me put it here. This was a really cool paper. If you can get this paper, it's a 2021. It says detect thy family, mechanisms, ecology, and agricultural aspects of kin recognition in plants, which is fascinating. It's really, really cool. So yeah, there is a lot of papers on it, but they're still working on it. More research needs to be done.
Right. Yeah, I have two thoughts about that. Yes, it probably is important for non-science people maybe to give some human attributes to things that aren't human because it makes them more relatable. So it's not all bad. But to me, it's a little off-putting because I think things are so subtle. The world is
Mm,
chemistry,
mm,
and plant communication is chemistry. They don't exactly have a nervous system, although some people say, plants are sentient. Well, I don't know about that. Anyway, there's that. Yes, a personal aspect gives people relating more to what's out there.
mhm.
I forgot my second thing. Okay, inoculation with mycorrhizae. Kathy Cripps has papers. And I think if you go to, I mean, the literature about mycorrhizae out there is overwhelming. And if you would just Google inoculation, and then do you want ecto or endo kinds of mycorrhizae, you could probably get information on it. Yeah. You need a greenhouse and you need patience, because as Kathy said, it takes 1 to 3 months to get her pine trees real well inoculated with the swillis.
Sharon,
So,
oh,
okay, pardon me.
oh, if you sorry, I didn't mean to interrupt.
So I'm looking at evidence of mycorrhizal connections between trees and nearby shrubs and forbs. Well, yeah, Suzanne Simard said, yes, there was a connection between the pines and alders, if you consider alder a shrub. Something like a water birch. I don't know, is that more of a tree or a shrub? It kind of depends on how it grows. Yeah, there was a mycorrhizal connection. Now, I'm thinking about my backyard. Let's say between a chokecherry and an aspen, maybe. Between a chokecherry and a pine, you'd have to go in the woods and examine it. So, a lot of these studies say, yes, there is relation. There are connections between things in nature, trees and shrubs. Now, with forbs, I did something bad. Most forbs are endomycorrhizae, and there's a lot of evidence that says no, there's not a lot of connection between endo and ecto mycorrhizal plants. Am I still on?
You are still on.
Okay.
Yes.
Okay. Oh, the name of the book. Well, I don't have, can you go?
Oh, yeah, the Arbornaut.
Right. Arbor knot.
I'll put it in the chat.
Okay. Yeah. The arbor knot. Yeah. So, yeah, there's as I say, she wasn't original with that, but they're starting to build all kinds of places. In the last part of the book, she talks about going into India, where they're making big pathways in the high jungle forests to find out what's at the very top of those jungle type plants. Yeah.
Mm.
And Washington and Oregon and Northern California, they've been doing that with the big redwoods. Yeah.
Mm. It looks like Beth has a question for us. Beth, do you want to add something to the conversation? Beth McFawn.
Okay.
Hi, yeah, thank you. I tried to put it in the chat, but it didn't work. But as a landscaper, I had a question. Using imported soils on a job site and planting trees and shrubs and perennials, I just wonder if there are additives that we can use to increase the mycorrhizal fungus.
Well, you can buy mycorrhizae. There are supply houses that sell them. As I said, when I was doing my reading, and I think it's Kathy Zabinski at MSU who says, the evidence isn't there to say, yes, you can inoculate and it'll make things better in a horticultural system.
Mm-hmm.
The idea is to inoculate the plant evidently before it's planted, like what Kathy Kritz does, and what they did with the ponderosa pines for eastern Montana, to just add mycorrhizae to the soil. I think the evidence is not there. for whether it's good or bad.
Okay.
One conflict, I mean, it depends on what soils you're working with. And remember, if you fertilize something, chances are you're not going to get mycorrhizal connections. On the other hand, something has to be healthy enough to have mycorrhizal connections. So there's a balance there. You know, thinking about my own backyard, most of my trees do well, but I do have one tree that one pine tree that I always need to fertilize. So it just needs that extra boost. I haven't dug up the roots, but my guess is it's probably mycorrhizal because it's next to a ponderosa pine. So I think I'm hemming and hawing because it's the usual, it depends kind of deal.
Excuse me, did you just say that fertilizing is not beneficial to building mycorrhizae in the soil? Or
Correct.
does it depend
Correct.
on the type of fertilizer? If it's like an organic,
Well, the idea
and not a
is,
chemical compound, I guess what it's derived from.
I think it might depend on the seasons and it might depend on the conditions. Here's the thought about fertilizing, that if you fertilize a tree, it's going to get everything. If you fertilize it really well, it doesn't need to spend its sugars. I mean, mycorrhizal association, there's some expense involved in feeding that fungus. So if it doesn't have to feed a fungus, if you fertilize it, it doesn't need that mycorrhizal connection. But in nature, things aren't fertilized. So they need that mycorrhizal connection. So here's the balance, that in a yard, let's say, to get something established, you might need to fertilize it to get it healthy and growing well, make it healthy, and then back off, because if it's stressed, then maybe it will feel the need to make a mycorrhizal connection, especially if there's a similar tree next to it. So there's the balance. It has to be healthy enough to make the mycorrhizal connection, but if it's too healthy, it doesn't need the mycorrhizal connection. So that's the balance, I think, in horticulture. What I have learned is that some nurseries are inoculating their trees and bushes with mycorrhizae before they sell them. So that gives them a good start.
Okay, thank you so much.
We did have someone ask in the chat how you got a ponderosa pine to grow in your yard, and Robin responded that she has several ponderosa pines in her yard, but would you care to speak to that?
Yeah, one I bought from a nursery a long time ago, and it's in one corner of the yard. And in another part of the yard, I was out in eastern Montana in the Custer National Forest. Actually, my husband was, and he dug up a little seedling and brought it home, and I put it in my yard, and it grows just fine. So naturally, ponderosa pine does not grow so well. You don't find it in the Yellowstone ecosystem. But you could plant it and it grows. The one that I planted with the soil from the Custer National Forest grew better than the other one.
Interesting.
Now they're both huge and they're fine, but yeah, it's a strange thing about ponderosa, this state tree of Montana that doesn't grow in the Yellowstone ecosystem. It isn't found naturally.
Right.
Yeah, if you plant it, it grows.
Would anyone else like to add to the chat before we wrap up for the evening, or ask a question directly?
Thank you, everybody.
Robin. Yeah, thank you everyone so much. And thank you, Sharon, so much for sharing this evening.
Thank you, Sharon.
Thank you, everybody.
Thank
All
you.
right. Oh, we have Karen. Yeah, go ahead. Karen Porter, you're welcome to unmute if you have a question or a comment.
Okay.
Am I unmuted?
Oh, yep. Now we can hear you.
Sharon, I just wonder what what your thoughts are on what we know are the very extensive operations to reseed the white bark pine. And we've all seen pictures of the little potted sprouts in their little pots with what looks like ordinary potting soil. Do you think those are being treated with mycorrhizae?
The ones that Kathy Cripps is working with, yes, they are. She
Good.
grows them in the greenhouse, and as I said, she says it takes 1 to 3 months to make
That's the one.
yeah, a good mycorrhizal connection. So she's been working with the Forest Service. I think it's the Forest Service on that. So yeah, that's going to give little seedlings the best chance to just pull them out of a backpack and bare root
Yeah.
them. Just lots of luck.
So when those finally do get planted, they're going to go into soil they've never seen before.
Right. But if they have the fungi with them already, they will have a head start on being able to get the nutrients in the water from the soil.
Right. So that's an indication of how critical those mycorrhizae really are.
100% critical. Oh yeah.
Yeah.
Oh yeah. Yeah. So I don't know where she's planting them. I don't know if this is a Yellowstone Park thing or if it's a Forest Service thing or both. But that's the best chance for planting seedlings. Unless I can think of an exception to that, unless you harvest log 1 year and plant the next. So you still have those fungi present in the soil.
Right.
But if you wait too long, you're going to lose those fungi. So yeah, either replant right away close to, as I say, big trees, or inoculate. Yeah.
Thanks.
And Sharon, one more question appeared in the chat. Kate Myers is asking, do diseases transfer along the fungal connections?
Actually, unfortunately, there does seem to be some evidence for that some pathogens, yes, can be transmitted. Yeah. Everything can be transmitted, but the benefits will way outweigh the harmful parts.
All right. Well, thank you so much, everyone, again, and we make sure to keep an eye out. Mark your calendars for our next for the 3rd Thursday of December for our next speaker, and yeah, we look forward to seeing you soon. Take care.
Okay, thanks, everybody.